and welcome to this episode of Skincare Confidential, a podcast dedicated to giving you a 360-degree view of the skincare industry. My name is Dr. Ted Lain. I am a board-certified dermatologist. I am the host of today's episode, and I'm also a co-founder of the Science of Skincare Summit, which is an educational conference, again, dedicated to skincare, occurring September 21st through the 23rd of 2023 in Austin, Texas. But enough about me. Today, we have an amazing guest for our podcast, Dr. Rahul Mehta, someone who I consider a friend, a colleague, and someone who I turn to when I just don't understand anything about skincare. He really sets me straight. So, Rahul, thank you for coming on and being our guest today.
Oh, you're welcome. Thank you for this opportunity.
Okay, so Dr. Mehta is the Vice President of Research and Development at Allergan Aesthetics. He actually started at SkinMedica before it was acquired at Allergan well over 18 years ago. Currently, his job duties include lead product development, in-licensing, due diligence, global regulatory, and global medical affairs functions for SkinMedica and the Diamond Glow brands of Allergan Aesthetics. He's leading the global expansion of Allergan Aesthetics skincare portfolio, and he established a skin quality and aging research center to streamline discovery, development, and assessments of skin quality related research. He is a busy guy. So, Rahul, please, how did it all get started for you? I mean, it looks like you actually were in pharma before you got into skincare.
That's right, Ted. So I'm a pharmaceutical scientist at heart. I got my PhD in biodegradable drug delivery systems, working on cancer chemotherapy, working with biopolymers like polylactic acid and nanoparticles and liposomes. And then I continued my research in academia. Actually, I was on the faculty at University of Kentucky, working on peptide and polymer and protein delivery, sustained release, and things like that. Then switched to industry and spent another 10 years working on antisense oligonucleotides. So, these are small modified DNA molecules that bind to messenger RNA and stop protein production. And the targets we are going after were really oncology and immunology indications. So one of the last project I had in the company was looking at topical delivery of antisense oligonucleotides. And we are going after psoriasis using TNF-alpha and ICAM and VCAM and other targets like that. So that was my introduction to the dermatology space.
Wow.
And then 2005, I joined SkinMedica and really started to apply my pharmaceutical expertise to elevate the formulation design and the claim substantiation processes for skincare. As you know, those are the key that takes up a lot of resources. And that's where we need the most help to elevate really the skincare products to the activity that we really expect from them.
Amazing. So you joined SkinMedica, just to go back in time. You joined SkinMedica. It was Dr. Fitzpatrick's company at that time, correct?
Absolutely.
And not only a skincare company, but also a med derm company as well. Multiple topical medical dermatology products, right?
Absolutely. And that's how I got my foot into the door, because I was part of the medical derm unit at SkinMedica, and was very fortunate to work with Dr. Fitzpatrick for the first 7 or 8 years. And he basically guided me in terms of what type of products he wants for his patients. But then the skincare products were so interesting, and I couldn't understand them really well because I was putting my pharmaceutical lens to these products and say, why are they working? They are not supposed to work in the way they are formulated. So I started getting more and more interested into the skincare and trying to really get into why some of the products works why some don't work, and got excited about the TNS that was part of the franchise at that time.
Yeah. So you actually came in while TNS was being developed or post-development?
It was already launched. The first product was launched, the TNS Recovery Complex, which was the growth factor product by itself. And then we developed the subsequent products after that.
Right. And since that time, obviously, you've helped to launch multiple products in the SkinMedica portfolio, including some most recently that I'm really excited about, and we'll get to later. But talk to us about the TNS Advanced serum, just that next generation of that TNS that growth factor serum with peptides and antioxidants that's kind of the bedrock of the SkinMedica portfolio.
Right. So just stepping back just a little bit on the TNS itself, what
Yeah.
we used to call a growth factor product, what we now know is that it's not really a growth factor product. It really is a whole fibroblast secretome. And by secretome, we define everything that the fibroblast cells are capable of making when they're activated. So that's what TNS really is. And when we launched it back when Dr. Fitzpatrick launched it, the claim was it has 7 different growth factors, because that's all we could analyze for. What we now know is that it has literally thousands of different molecules like proteins, peptides, and they have different functions, and and also large number of exosomes. But what really differentiates that original TNS and this next generation TNS that we launched, we call it MRCX, is how the secretome is derived. So in this new product, we actually grow the fibroblast under hypoxic condition. So under
Mm-hmm.
hypoxic condition and low gravity, by suspending them, and the fibroblasts really start take up characteristics of not necessarily stem cell, but there are more stem cell like properties in fibroblasts. And so the secretome of these modified fibroblasts now contains actives that are more emphasis on inflammation, stemness, versus original TNS, which was more uh, uh, actually emphasis of the product was more on extracellular matrix rebuilding.
Okay.
And these are all in vitro profile, of course, not really
Sure.
clinical profiles.
Sure. So that's so interesting. Okay. So let's just go back and define some terms because not everybody listening understands what a secretome is. So my understanding is a secretome is the gestalt that when you look at everything that is made by a fibroblast, which are those cells in the dermis that are responsible for collagen production, for example, and most of the components of the extracellular matrix. If you think about the dermis, it has these fibroblasts that are kind of suspended in a gel or a liquid that is comprised of not only collagen but hyaluronic acid and multiple
Right.
other glycosaminoglycans, which are those
Hmm.
chemicals that give the skin turgor and firmness. And
Yes.
the fibroblast cannot, the importance of the fibroblast, first of all, for anti-aging cannot be understated, right, Rahul?
That's right. Yes, and when we say secretome, it is everything the fibroblast is secreting out into this extracellular matrix, and the secreted components have their main function is to communicate with other cells. Other fibroblasts communicate with keratinocytes, communicate with cells of the immune system, and specifically when skin is under stress or when repair is needed, fibroblasts communicate to these cells that we need to introduce whatever it takes to induce the repair and regeneration of the skin. So secretome contains all the messages
Mm-hmm.
that the fibroblast is trying to send to the surrounding cells.
Yeah, this is so interesting because for so long people were looking at TNS serum and we knew that our patients were getting improvement with TNS serum, right? We
Yes.
saw the improvement and we have so many patients who would come to us and say, I love this product. I use it twice a day. It absolutely keeps my skin feeling and looking better. But we never really understood how it worked because we know that these growth factors, which was really the story for so many years, are just too big. They're just large molecules, and it's difficult in my understanding, in my very primitive understanding compared to you, Rahul, in terms of how they penetrate through the barrier. And so now, with this whole secretome idea, and you said the magic word of exosomes, perhaps now we're really understanding that this is more of an anti-inflammatory messaging compound than true delivery of growth factors.
So yes, you're right, and again, that was one of the biggest question I had when I joined SkinMedica and started looking at growth factors. My my first question was, how do these go through the skin? First of all,
Mm-hmm.
how are they even stable at room
Mm-hmm.
temperature?
Mm-hmm.
And then if they're stable, how do they go through the skin? We never got answer to how they go through the skin, but we do know that they're stable. The way Dr. Fitzpatrick and the formulators created the products, the growth factors in that and all the proteins and peptides are stabilized in the product. But we could never answer the question of how what's going through the skin that's actually working.
Mm-hmm.
I knew growth factors are too large to go through the skin. There's some mechanisms by which skin can actually sample large molecules, like a typical allergic reaction, right? You get large protein on the surface, they transmit some response into the skin and you get the allergic reaction.
Mm-hmm.
But those are really rare and really small amount of things go through. So we didn't really know what was working until few years ago. We heard about exosomes. We didn't know what they were 20 some years ago. And we now believe that exosomes in TNS can actually penetrate the skin and provide the biological activity uh, that we see in all of our in vitro model, and that could translate to clinical activity.
Okay, so let's dive into exosomes for a second. Exosomes are really carrier molecules. Let's talk. They really
Yeah,
are liposomes in the way I think of them, and they can contain many different ingredients or skincare beneficial products inside of them. The problem is, as you and I have spoken about, how do you stabilize a liposome
right.
in a skincare product? And let's talk about that, because from my understanding, that's really the secret sauce to TNS's efficacy.
Yes, and so if you recall, the TNS product is dual chamber. We have all the growth
Right.
factors and exosomes on one side, and the rest of them on the other side. That's the best way we could actually stabilize right now, because we know that exosomes and even the peptides and proteins, they cannot be come in touch with surfactants or oils or everything that's typically present in a formula. They're similar to liposomes. Liposome break down as soon as you put them in an emulsion or have a
Mm-hmm.
surfactant on oil with them.
Mm-hmm.
Exosomes are no different. They're lipid lipoprotein membranes
Right.
that break down very easily. We do know that the formula of TNS recovery complex with the high protein concentration and the actual composition, the exosomes are stable for at least 4 years, because we have isolated exosomes and they show biological activity. Uh, it's a challenge when we extract them further, and if we want to put them in a product mixed with other things, that's the challenge that we're working on right now.
Mm-hmm.
And there are ways to do that. It's just it's very hard to get something that is stable at room temperature, because you can't really have a skincare product that you have to refrigerate. It doesn't work practically. And then you do want a 2 year shelf life. So those are the questions we are answering right now for exosomes themselves. But as
Mm-hmm.
far as the product itself concerned, the exosomes in it are stable and they're being delivered right now.
So, Rahul, as we compare different companies are producing different exosome products, right?
Hmm.
Some of them are adipose stem cell
Yeah,
derived exosomes. Some of them are plant-based exosomes. Some of them are platelet derived
yeah,
exosomes.
yeah.
For SkinMedica, then, are we saying these are fibroblast derived exosomes?
That is correct. Yes. So
Okay.
uh, the way exosomes work in terms of transferring message, fibroblast exosomes, their main function is to maintain skin health. And so
Mm-hmm.
the messages from fibroblasts that are in the exosomes, whether they're all the microRNAs that are in there or the DNA fractions or
Mm-hmm.
peptides, they're
Mm-hmm.
designed to maintain skin health and skin homeostasis. And
Mm-hmm.
so I believe that fibroblast-based exosomes are the best for skin, because that's what they normally do.
Makes sense.
Now, exosomes from other sources, platelets, they're extremely important in wound healing, because that's platelets function. So exosomes from platelets have a big role to play in early stages of wound healing, that may translate to maybe some anti-inflammatory action, or activities like that are early part of the wound healing process. Uh, Exosomes from adipocytes, again, their main function is to maintain adipocyte health.
Mm-hmm.
And so they will be good at that. There are other exosome sources like the bone marrow exosomes, uh, and the bone marrow stem cell exosomes. Their main function would be to help grow the blood cells. And so I personally believe that for skin regeneration and maintenance of healthy skin, you need exosomes from or whole cell secretome from fibroblasts, which
Yeah.
are which is their primary function in the skin.
Yeah, that makes a lot of sense. And I guess this really correlates with the development of the Skin Quality and Aging Research Center, right? I mean,
That's
as
correct.
you are, and which I understand is just launching in the last year, and really there's a new person that's going to lead this along with you to streamline the discovery of these new ingredients like exosomes. And I'm sure, given the resources that AbbVie brings to bear on Allergan's ability now and SkinMedica's ability to find new ingredients, now we really have the resources necessary to kind of propel SkinMedica to the forefront.
Absolutely. And again, when I see the activities we used to do 15 years ago or even 10 years ago, the resources we had and what we could do with that versus what we can do now, it
Hmm.
really shows the power of pharmaceutical research engine behind SkinMedica. The whole Allergan Aesthetics has a big discovery team and a big biology team that helps us. But going beyond that to AbbVie has thousands of people that we actually work very actively with. That Genomics Institute is helping us create uh, kind of uh, getting into completely new era of drug discovery. And along with drug discovery, as we discover new targets, we are also going to apply them to skincare.
Yeah, well,
And
there you
that's
go.
really the main function of the Aging Research Center.
And I think we can kind of start talking now about the even incorrect system with the lotus sprout extract, which I'm really enamored with. I think, from my understanding, again, Rahul, correct me if I'm wrong, but you really did take that drug discovery process and the assets that are available to you to really find this new ingredient, which has it really shown to reduce pigmentation.
Absolutely. And this is the first example where we actually took something that the Allergan facial aesthetic discovery team was working on. They discovered a key molecule that had a significant effect on pigment, reducing pigment. So the SkinMedica biology team and the SkinMedica formulation team, working with the Allergan discovery team and our large number of cosmetic ingredient industry partner, we identify a natural source for these active molecules. And what
Mm-hmm.
we discovered is that a lotus plant has these molecules, and specifically in the sprouting stage of the seeds, has the highest level of these active molecules. And so, working with these companies, we developed process to extract what we call non-lotus sprout extract uh, that contains the highest level of these two actives, which we'll be publishing soon.
Okay.
And so we also then discovered, working with a very large academic institution, that these molecules actually work by a pathway that was not used before in skincare, which is really enhancing melanosome autophagy. And so the lotus sprout extract actually increases melanosome autophagy in keratinocytes as well as melanocytes. So what happens? The melanosomes that are already migrated into keratinocytes, that are in the sitting on top of the melanocyte layers, lotus sprout extract is able to activate keratinocytes to basically digest those melanosomes and remove the melanin from them. And
So
so
so it activates the keratinocytes or macrophages?
it actually activates keratinocytes. So
Okay.
so so we are not removing melanocytes. We are activating the cell's internal processes of autophagocytosis. So macrophages are
Ah,
not included here. They are not
okay.
involved. But internal pathways of keratinocytes are actually increasing the autophagy of the melanosomes that are inside the keratinocytes. And
Okay.
so
So they would then be encased in essentially a liposome as well then and expelled from the cell. Am I thinking about it right?
I think they get encapsulated into lysosome and get digested, because if
Just
they're
digest.
expelled out, they'll still be in the skin,
Yeah.
but they're actually enzymatically degraded, and
Okay.
and we are actually continuing the research to understand that better as to how is that happening.
Okay. Interesting. So yet another pathway for brightening and lightening the skin.
Yes,
I think most recently we've thought about, another company has come out with peptides and a peptide complex to help lighten the skin. And before that, we were really focused on tranexamic acid, which focused
yes.
on the inflammatory pathway of melanosome production. And so now yet again, we are using the scientific discovery and the drug discovery method to find another pathway that we can focus on for lightening of the skin, which is, quite frankly, especially here in the South where I am, just the most important responsibility for skincare products.
Absolutely. And again, pigmentation is probably one of the most difficult condition to treat, because if you think about it, melanin is essential for life.
Mm-hmm.
And so cells would do anything to let you remove melanin. It's not going to let you remove melanin that easily. And that's
Yeah.
why we need all the different approaches. Tranexamic acid works great. We have a lot of peptides from a lot of other companies that also work really well.
Mm-hmm.
What we need is to manage as many different pathways as possible, so we can get a product that works fast and is something that can be used in all different skin types.
Well, and if you think about the even incorrect system, which is a system of three different products, right?
Right.
You've got the kind of spot correcting product, which has the highest concentration of that lotus sprout extract.
Yes, it's a three product system. The main serum contains the highest concentration of lotus sprout extract. It also has what we call LTN complex. So lotus sprout extract, tranexamic acid, and niacinamide. And this is a
Okay.
product where again it comes to the resources that AbbVie is able to provide us. We spent almost a year optimizing penetration of tranexamic acid into the skin.
Wow.
That went into this product. So by combining these three, we are able to achieve. In fact, the clinical studies we are doing right now, where we use the pad for exfoliation and early washing,
Right,
followed
which
by
that
the
pad, by the way, sorry to interrupt, but I think we're excited about that pad, which is an alpha hydroxy acid plus a retinol in
yes.
that pad, right?
No, the pad contains lotus seed extract as well as tranexamic acid and glycolic acid. The spot
Okay.
treatment contains retinol
Oh,
and glycolic acid as well as the 3-LTN complex. But
okay.
when we use all three together, we are seeing results amazingly fast. Within a few weeks, we start seeing changes in skin, which I wouldn't have believed if I didn't see it in the number of patients that we have used it on.
Yeah. And, I'm honestly, the pads with the combination of products, which I think is quite unique. I have not seen a pad skincare product with that combination.
Right.
In my practice, we're all kind of excited about it for our acne patients as well, because you
Yes.
can imagine the
Yes.
glycolic acid will be exfoliation, but then the post acne blemish marks will be lightened. I'm excited
Right.
about that with that lotus sprout extract. So I think
Exactly. Yes.
you'll see that product just as a standalone really explode as people realize the benefits for going beyond just your
Right.
standard PIH or hyperpigmentation or melasma. It's going to be used for other stuff.
Right, and we'll I mean, our philosophy is that we continue doing studies. So we
Mm-hmm.
will continue studying this product in other indications like acne and everything. With once now that the product is launched, we'll continue publications on these other indications with the
Mm-hmm.
products.
But there are other products that have the lotus extract, right?
Yes,
This
yes.
this is not your grandma's lotus extract though.
No, in fact, when we that was part of our kind of due diligence. We looked at all the different lotus seed extracts and lotus plant extracts that are out there commercially available, and analyzed them for these two actives that we were looking for. And in fact, none of the other extracts actually contain these actives, because
Okay.
the way the processes are developed, they are removed during early stages of purification. And so we had to develop a process specifically to retain those two actives. And so the other products in the market do not contain the actives that we have in the lotus sprout
Proprietary.
extract.
Okay, proprietary to Allergan's product
Right.
line there.
Yeah.
Okay.
Yeah. Yeah.
Now that this product obviously is going to be further studied in the Skin Quality and Aging Research Center, but
Absolutely.
as you
Yes.
look, I'm sure there's other kind of broad topics that are really interesting to you since you founded this
Mhm.
center. When you look at the next few years, what do you think will come out of this Skin Quality and Aging Research Center?
So I think this is a long-term project, right? So
Mm,
so so we are focusing on hallmarks of aging. So we're looking at mitochondrial health. We are looking at senescence, telomeres, all those at the basic cellular level. And I'm hoping that in 8 to 10 years we would have new targets,
mm,
that a completely new target that we can't imagine right now, that can help us with skin aging and the skin quality changes. And other thing we want to focus on during this time frame is also diversity biology. We know different skin has different biology. We are not taking advantage of that when we create products. And
just so
so
true.
so we'll
That's
we'll
very
focus
true.
on that. Little bit shorter term, 3 to 5 years, we'll probably see more personalization. um, uh, it started, but there's still a lot to learn from the AI perspective when it comes to personalization. We are just not there yet, but personalization based on genetic profile, epigenetic profile, microbial assessments. Some of these things you start seeing, there's some companies that are doing good work in this area, and you start seeing products personalization. But really, what we need is a huge biobank type repository where we have 500,000 people, where we know their genetics, we know their epigenetic profile, we can follow them longitudinally, and then we know their skincare needs and skincare conditions. So we are that's one of our other initiative, the Skin Quality and Aging Research Center, is to identify a biobank or create a biobank with AbbVie's resources that can help us identify different groups of people and what their skin needs are, look at their genetic and epigenetic markers, and see if we can find new targets that we can engage in discovery.
Imagine that, right? If you could tap into ancestry.com or one of these other large, DNA biobanks and really, use your AI learning neural networks to come up with some really interesting solutions.
We will get there. These are long-term projects, and so hopefully before I retire, we'll see something like that.
Well, if you need any DNA, I'll donate mine, Rahul.
Thank
what
you.
do I need?
We'll
Just a little
take
cheek
you
swab?
up on that.
Yeah, be careful what you look for.
Yeah.
Okay, great. Listen, Rahul, thank you so much for this wonder. I mean, I learned a ton. I always learn a ton talking with you, and I so appreciate your time. We are so excited about the new direction, the new developments coming out of SkinMedica, and I think the future is incredibly bright. Not to mention, we haven't even had time to really talk about the Diamond Glow, which is the device to, that has serums that can, increase penetrations of active serums, which will further development will be supporting that device. And I'm sure we'll see some more data coming out from there as well, knowing your team.
Absolutely. There's a lot to come.
Produced. So. Yeah, always being produced. So thanks, everybody, for listening to this episode of Skincare Confidential. Don't forget to like. Don't forget to subscribe. Don't forget to refer us to your friends. If you have any questions about skincare, if you have questions about skincare products, or you'd like to learn more about ingredients, shoot us an email at info@scienceofskincaresummit.com. And you never know, maybe your question will be answered at the next podcast episode. This is Dr. Ted Lain, and with me is Dr. Rahul Mehta from SkinMedica, signing off. Thank you.